In this section, let's now turn to how could we estimate a population average treatment effect if we wanted to. As a statistician, I will say that it's always best to try to do a better job through trial design. That sort of statistical methods can only help us so much after the fact, and that if we want to estimate a population average effect, we should really be thinking about that from the beginning. So the ideal scenario will be random sampling, where we have a population of interest, we randomly sample from that, and then we randomly assign treatments within that sample. This is great from a statistical perspective for estimating a population effect, but it is quite rare. I know of maybe a handful of examples. Almost all of these are evaluations of large government programs, often administered in sites, where the government could basically sort of enroll all of the sites or a random sample of the sites, and then randomly assign participants within those. Examples include the Head Start Impact Study of Head Start programs, as well as Upward Bound, which is a program for college students, where again, there were sort of upward bound centers, so they could enroll a random sample of centers, and then within those centers, randomly assign people to get into the program or not. I do think this is an area that we could do more. So for example, health systems could use their electronic health record system to randomly sample patients for potential enrollment in trials, of course, with the appropriate consents obtained. But in cases where we have population data, such as from an electronic health record system, I think we can be more clever and thoughtful about how we use that to gather information and enroll the people who are relevant for some research question. A second example is what's called purposive sampling. So this is an approach where a researcher might say, well, I know I want some small schools and some large schools, and I want some urban schools and some rural schools. And so kind of approaches the sampling or selection process, not in a sort of random sampling way, but just being thoughtful about trying to reflect the spread of sort of types of settings or people that might be relevant. This doesn't, though, give the formal representativeness and unbiasedness that the random sampling gives. A third approach, and this is one that has become increasingly popular in the field of mental health research, are what are called practical or pragmatic clinical trials, which are basically studies that are very large scale and aim to enroll a broad spectrum of people and clinics. So rather than, say, just recruiting from Johns Hopkins and Harvard and Stanford, they really go out into a broader set of places and really try to enroll, maybe centers and sites across the country in a range of areas, and then enroll patients across those. These are potentially quite useful for this population average treatment effect purpose, but they are very expensive and require significant resources to do them well. But fundamentally, again, the main idea for these would be to select subjects for a trial in a particular way, and at least be thoughtful and upfront about who is being included and who might not be. So now let's turn to sort of post hoc analyses. And I'll first talk briefly about scenarios where there might be multiple studies available. So many of you might have heard of meta-analysis, and the idea here is that maybe there are 10 different randomized trials on some new surgical tool. And so the idea is that if we combine all of the data from those trials, that sort of the overall results might reflect a population effect better. And I think that's sort of one of the underlying goals of meta-analysis. But the problem is that in general, this has not been an explicit focus of meta-analysis. A focus on populations has not been part of it. And of course, the main challenge here is that combining a set of non-representative trials doesn't necessarily give you representativeness. So if all of the trials sort of were conducted in the same or similar non-representative populations, we might not be getting any closer to the target population that we are actually interested in. So meta-analysis might be helpful, but doesn't necessarily solve this problem. Another approach, which is a really interesting area of ongoing statistical work, is known as cross-design synthesis or research synthesis. And the idea here is to combine both trial results and non-experimental studies. So maybe we have a couple small non-representative randomized trials, But we might also have a non-experimental data source with non-experimental estimates about the effectiveness of this treatment. And so cross-design synthesis sort of brings together the randomized evidence and the non-randomized and accounts for the different populations that these might reflect. So that can be a very compelling approach if you have that sort of data situation. So unfortunately, in a lot of the fields that I work in, we don't have the luxury of multiple studies, though. Often we just have a single study, maybe a single randomized trial, and some data on a population of interest. So I will briefly talk about approaches here, and then we'll go through a case study. So one of the sort of potentially obvious approaches that some of you might be thinking about is what's known as post-stratification. And the idea here is that, let's say we have a randomized trial, and we know that the age distribution in the trial does not reflect the age distribution of our population. So what we can do is we can estimate effects separately for subgroups defined by age, and then basically reweight those using the population age proportions to get an overall population effect. So that will sort of upweight the groups that maybe were not represented in the trial and downweight the ones who were overly represented in the trial, and sort of rebalance the age distribution to match the population. So this can work really well if you have, one variable that you're worried about, like age. But often in these contexts, we might be worried that our sample and population differ on a number of characteristics that might moderate treatment effects. So in this case, weighting can be used as a way to sort of smooth across a number of these variables. So if any of you are familiar with survey sampling or non-response weights or propensity score weights in non-experimental studies, the idea here is the same. Basically, we can model the probability of participation in a trial as a function of a set of baseline characteristics, and then weight the trial members by their inverse probability of participating. So people who were sort of, again, underrepresented in the trial, so they had a low probability of participating, will receive a higher weight. On the other side, people who were sort of overrepresented in the trial, they had a high probability of participating, will be downweighted. And doing this weighting will then sort of make the sample look like the population on the set of factors used in the weighting. And we'll talk a little bit more about this. So that's one approach. Another approach for estimating a population average effect is basically to use the trial data to fit a flexible regression model of the outcome as a function of the treatment status and a set of covariates, baseline covariates. So the idea is to, again, sort of fit a flexible regression model where you can then use that to predict outcomes in the population under treatment and under control. One particular way of doing this is using an approach called BART, Bayesian additive regression trees. And that can work quite well for this purpose because of, again, sort of the flexible way that it models the outcome as a flexible function of covariates. There also are a set of doubly robust approaches that combine these weighting and outcome model-based strategies, and these can be excellent approaches. We'll talk a little bit more about these in a case study, and as I alluded, this is also a growing area of statistical research. I want to step back for a minute just to sort of be clear about what the presumed data structure here is. So the idea here is that we have a randomized trial, which is the top part of this table. So in that case, S equals 1 for trial membership. And in that trial, we observe outcomes, treatment status, and a set of covariates. We also have a population data set, so information on our target population of interest. S equals 0 for that part of the data because they were not in the trial. We don't observe outcomes or treatment in the population. The sort of premise is that the population doesn't yet have access to the treatment. But we do observe these covariates. And for these approaches that I mentioned, the weighting and outcome regression approaches, you basically implement them by stacking the data kind of like this. So we will have a combined data set that combines the data from the randomized trial and from the population of interest. One key point is that these covariates need to be measured consistently across these data sources. And as we'll turn back to, this is often one of the challenges in implementing these methods in practice because often the covariates are not measured consistently across data sources, which makes it more challenging to use them directly. Let's now talk through a case study of what this can look like in practice. This example comes from a paper by Issa Dahabra that used data from the coronary artery surgery study called CAS. This was sort of a unique data source because it was a randomized trial that was nested within a cohort study. So we have the nice situation where we do have comprehensive data on a target population of interest in the cohort study. And then we have comparable data on people who agreed to be randomized sort of within that cohort. The trial itself was comparing surgery plus medical therapy to medical therapy alone for patients with chronic coronary artery disease. About 800 patients were randomized, and about 1,300 in the cohort declined to be randomized. The outcome of interest is 10-year risk of death from any cause, and there is a set of covariates available, including things like age, disease severity, and other measures of coronary health. So the idea here is that we have the randomized trial effect estimate, but we want to use that data and the cohort data to sort of project out what the effect of this surgery plus medical therapy intervention is in the full population, in the full cohort. So sort of generalizing from the 780 patients in the trial to the approximately 2,100 in the complete cohort. And what we did here was to use that weighting approach, weighting the people in the trial by their odds of participating in the trial to essentially make the trial participants look like the full cohort. One thing that maybe is reassuring though, although it doesn't make it a great teaching example, is that it turns out that actually the trial participants in this study didn't look that different from the non-participants, from the people who declined to participate. I would posit though that it's always important to at least check this when you have population data available, is to actually sort of start to investigate, well, who did enroll and what did they look like compared to the people who didn't? So what this figure is showing, the left-hand side of it, is showing the probability of trial participation. So this was estimated using the covariates I mentioned on the previous slide. And we can see here that the solid line shows the trial participants' probabilities of participation, and the dashed line shows the non-participants. So here we see that actually these are quite overlapping, and it doesn't look like there are substantial differences in the characteristics of people who did and didn't participate. The right-hand side of this figure shows the resulting weights for the trial participants. They range from about 1 to 6, not a huge range, and again, that's partly because we don't see huge variability in the probabilities of trial participation, and we don't see big differences between the participants and non-participants. We can also look at each covariate individually to help understand what our trial sample and population look like. And so here you can see in the non-randomized group, which did actually some received surgery and some only received the medical therapy, we can see that sort of their age was around 51, angina percent was around 80% across these, etc. And then on the right-hand side, we see the randomized group, the people who agreed to be in the randomized trial. And again, when we see sort of broadly similar rates for these characteristics. So again, sort of on these individual characteristics, we don't see much. And that's, again, sort of related to the fact that those probabilities didn't vary as well. So what we can then do, though, just to sort of check and estimate our population effects, is compare the results that we see from the trial itself to approaches that do try to adjust for these sort of minimal differences between the trial and the population. So in this case, if we look just at the trial and sort of forget our population data, the risk difference was -3, sort of a confidence interval that includes 0. If we then want to estimate the population average treatment effect using a few different approaches, that's what we see in the next few rows. We can do an outcome model-based approach, we can do this weighting approach, or we can do a doubly robust approach that combines both of these. And we see here that the point estimates of the risk difference are all a little bit smaller, but the confidence intervals are broadly similar to what we saw in the trial only. So you might notice, though, that the confidence intervals here are wider when we are trying to estimate the population effect. The 3 bottom rows of this table have slightly larger confidence intervals. And that's basically reflecting the fact that we now are trying to extrapolate from our trial sample to the broader cohort. So that's something you'll often see when using these generalizability methods. But again, in this case, not surprising, given the similarity in the covariates that we saw, we don't see a big difference in the overall conclusions in terms of the effectiveness of the surgery for this group. I want to be clear, though, that other examples might see differences, and that's going to depend, again, on sort of the extent of the effect heterogeneity and the extent of the differences between the sample and the population. I do want to take a minute to talk about the assumptions that underlie these approaches. Sort of what is required to interpret these population effects as being good estimates of the actual effects in the population. So first is what's called positivity. Positivity is an assumption that says that everyone in the target population had a non-zero probability of participating in the trial. And this sort of makes sense intuitively. For example, if the trial didn't allow anyone over 65 to enroll, then if our population includes people over 65, we basically are going to just really have to extrapolate from the younger group to them. So there might be a group of people that we really don't have direct information on from the trial. And so that's part of this assumption, that we sort of have to believe that the people in the target population sort of could have enrolled in the trial. The second big assumption is what's called ignorability. This is similar to an assumption called unconfoundedness in the non-experimental study context. In this generalizability context, what it's assuming is that all of the effect moderators that relate to sample selection are observed and adjusted for. In other words, that there are no unobserved differences between our sample and population on variables that also relate to the treatment effects. The challenge with this assumption is that it's often rare to have extensive data overlap between the sample and population. We often don't have extensive covariates measured consistently across the two. Because of this, we really need sensitivity analyses to assess robustness of the results to violation of this assumption. But if we're thoughtful about sort of designing our data collection, and if we align our trial data collection with population data, this can greatly help this by increasing the set of covariates that we can adjust for. So these are the key assumptions to think about when you want to use these methods, and sort of think about whether you observe the key effect moderators in the sample and population and whether they can be adjusted for.